Good evening, students. The other day we discussed the fungi general characters, thallus organization, and reproduction, that is in vegetative, asexual and sexual types. Today we will discuss the classification of fungi. Many mycologists gave different types of classifications, but now the following system is the Alexopoulos system. He placed the total fungi into mycota. Total fungi into a single division, mycota. The division mycota is subdivided into two subdivisions. One is Myxomycotina and Eumycotina. Myxomycotina are also called the slime molds. Eumycotina are called the true molds. Regarding characters, in Myxomycotina, the thallus is a plasmodium. Plasmodium is nothing but a plant body not having the cell wall. And they are holocarpic. We already know the holocarpic, the total fungus form into a single reproductive structure. Whereas in Eumycotina, the thallus is unicellular or multicellular, that is eucarpic. We have many reproductive structures you have. And regarding these examples, the Myxomycotina, Stimonatis and Physarum. Here the Eumycotina subdivision is further classified into 9 classes. It is divided into 9 classes. Those are Chytridiomycetes, Hypochytridiomycetes, Oomycetes, Plasmodiophoromycetes, Zygomycetes, Trichomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. This Deuteromycetes is called the imperfect fungi, but there is no sexual reproduction. So these are the classes that you have to learn, and let us see the characteristics of each class. The first one, that is Chytridiomycetes, where the thallus is multinucleate, oval and having unicellular thallus. Zoospores uniflagellate and A.V.V.S type. So in the diagram you can see this plant body having only one posterior side that is situated. Actually this one is anterior side, this one is a posterior side. So from this side only the flagellum is there. So it is attached at the posterior end. Example, Synchytrium. In Hypochytridiomycetes, this is a aquatic fungi. Zoospores having a single tin cell flagellum at their anterior end. At the anterior side, there is a single tin cell type of flagellum. The other day I told you the tin cell nature. Tin cell nature, that is a flagellum having a side branches, that is called tin cell type. So in this you can have the tin cell type of flagellum. Example, Rhizodiomyces. Rhizodiomyces. Regarding the third one is Oomycetes, where the mycelium is cenocytic. I think you know what is cenocytic. We already discussed in the thallus, that is a plant body, accepted plant body having a number of nuclei, or multinucleate accepted condition is known as cenocytic. And motile cells possess two anterior flagella. There are two flagella, anterior flagella, but interestingly one is a A.V.V.S type, another is a tin cell type, that you can find in the diagram, having two anterior flagella, among which one is A.V.V.S type and another is a tin cell type. The example, Saprolegnia. In the Saprolegnia, the zoospores having two types of flagella. The next class we have this Plasmodiophoromycetes. Plasmodiophoromycetes, the thallus is a multinucleate plasmodium and motile cells are biflagellate. Both are A.V.V.S type and anteriorly inserted but with a different length. Here it is very interesting. One flagellum is short and another flagellum is long. So this type of flagella are found in Plasmodiophoromycetes. Example, Plasmodiophora. The next class, that is Zygomycetes. Here the thallus is stenocytic or septate mycelium. Sexual reproduction by the fusion of gametangia. Two gametangia come together. We already discussed it in the last class. Two gametangia come together and they fuse and form a zygospore. This is called zygospore, is formed by the gametangial contact from two filaments. And very interesting thing we have to remember is the motile cells are absent. Example, Rhizopus, Mucor, etc. Next, Trichomycetes. Thallus is filamentous, branched and cenocytic. The members are attached to the digestive tract or the external cuticle of living arthropods. They are attached with living organisms and lead a symbiotic life. Asexual reproduction by a variety of spores and sexual reproduction by isogametangial fusion. This is also here two gametangia, similar gametangia come together and fuse and form the zygote. Example, Enterobris. This you can see an example. Okay, so this is associated with the animals. The next class is the Ascomycetes. Here the thallus is highly branched and having monokaryotic hyphae, and later it will become dikaryotic. So monokaryotic means a cell is having a single nucleus. After gametic fusion, where the cell becomes two cells, the cell becomes two nuclei, that is called dikaryotization. Sometimes what happens, the fusion of nuclei is postponed, and later only the fusion of nuclei takes place and form the zygote and form, we have haploid, that's what's called the haploid ascospores. Here the male sex organ is called the spermatia and the female sex organ is called the ascogonium. After plasmogamy, that is the fusion of cytoplasm, is known as plasmogamy, the dikaryotic mycelium develops. Ascospores are developed after garyogamy and meiosis. After that what happened? Ascus is developed. That ascus having two nuclei, those two nuclei fuse and a diploid nucleus is formed. Later the diploid nucleus undergoes reduction division and form haploid ascospores. Ascospores are developed inside the ascus. Ascospores develop inside the ascus. And spores are produced inside the sac-like structure, that is called asci. These are called endogenous. Endogenous means develop inside the cell, that is called endogenous. Asci are produced in fruit bodies called ascocarp. So this fruit body, here the motile spores are absent. Here the fruit body is developed and the fruit body is having of different shapes. You can find in Ascomycetes different shapes of these fruiting bodies. The other day we discussed that one is called the apothecium. It is just like a bowl-like structure. Example, Pezaiza. Another is called the perithecium, that is flask-shaped structure. Example, Claviceps. The third one, Cleistothecium. I already told the Kleistos means closed fruiting body. Example, Penicillium. In these two types, apothecium and perithecium, there is an opening in the upper side so that the dispersal of the spores takes place. Whereas in the Cleistothecium, after the decay of the outer wall only, the spores are released out, so that after finding a suitable substratum, they develop into that particular Ascomycetes fungus. So these are the three types of fruiting bodies found in Ascomycetes. The perithecia, it is a flask-shaped structure. Example, Claviceps. Another is called apothecia. It is a open cup-like structure. Example, Pezaiza. And this Cleistothecium, it is completely closed. This is called Cleistothecium. Example, Penicillium. The next class is the Basidiomycetes. The thallus is well developed, septate hyphae. There three distinct stages of mycelia are found in this. As soon as the basidiospore develops and give a primary mycelium, that is having a single nucleated cells, so called monokaryotic mycelium, and after the fusion it will become secondary mycelium. There you can find dikaryotic cells, and after the formation of the fruit body, there you can find the tertiary mycelium. Each basidium produces, here also you can find the fusion of the nuclei is postponed, and they fuse in the basidium and form a diploid nuclei, and then it undergoes a meiosis and form 4 haploid basidiospores, and these basidiospores are developed outside. Since this is a basidium, where you can find basidiospores are developed on the outside. So the development is endogenous development, and each one is known as a basidiospore. And sex organs and motile forms are absent in this agaricus. This agaricus fruit body, you can see now, agaricus fruit body, that is just like a mushroom, So this is in natural conditions. You can see the mushroom like this, and if you draw the diagram and label, where you can find the cap, this is just like an umbrella like structure, that is called pileus structure, and lower cells you can find a number of gills. Inside the gills you can find only the basidia and basidiospores, and this is the stipe. Stalk of this one is called stipe, and below you can have mycelial threads or rhizoid like structures for absorbing food materials from the substratum. Next form class, I told you already, this is Fungi imperfecti. Here the thallus structure and asexual reproduction resemble Ascomystis or Basidiomystis. That is, some forms show Ascomystis fungus like, and some show Basidiomycetes like. Here the sexual stages have not been discovered. Here there is no sexual reproduction. It carry only asexual reproduction. So they are called Fungi imperfecti. Example, Colitotrichum and Cercospora. These fungi also cause diseases. Colitotrichum and Cercospora. So next we will see the economic importance of fungus, economic importance of fungi. So here you can have the both beneficial as well as harmful effects causing the fungi. Now let us see the beneficial aspects. The first one, brewing and baking industry. So yeast ferment sugar solution into alcohol and carbon dioxide by the enzyme zymase. So yeast produces an enzyme zymase. That enzyme converts the sugar into alcohol and carbon dioxide. Alcohol is used in the brewing industry and carbon dioxide is used in baking industry. So I think it is you might have seen in houses when the preparation of dosa. See, generally the idli you can have a solid one, whereas in dosa you can find minute openings are there. Why they formed? Because while baking on a pan, what happened? Due to the heat, the carbon dioxide is coming out. When coming out, you will have a minute pores you can find. In the same way, you can find in the bread industry or baking industry also, the bread is very smooth because of the release of carbon dioxide from the material. You will have these porous and spongy like. So that is the effect of this carbon dioxide in baking industry. The next one, let us see the organic acids which are obtained from the different fungi. Different fungi gives different types of these alcohols. Organic acids. Fungi are used for the preparation of various organic acids such as citric acid. It is obtained from Aspergillus niger, and glucomic acid from Aspergillus niger. Here gluconic acid, N, this is here you have this N, gluconic acid, Aspergillus niger, fumaric acid, Rhizopus stolonifer, gallic acid, Penicillium glaucum, lactic acid, Rhizopus stolonifer. So the thing is, in botany and zoology you can come across the names are in Greek and Latin. So it is very difficult to write and remember, but that you have to practice to remember and write. Otherwise, you have to write once or twice. You can remember these names of the plants. Regarding the next one, the fungi as a research tool. So here, Neurospora, this is generally called the red mold, is used to study the loss of heredity and to understand biochemical pathways. That is, biochemical pathway. That is, I think the other day I told you already, one gene, one hypothesis, one enzyme hypothesis. There you can find the Inospiro, this Neurospora. You can study, this is Ascomystis fungus. You can study the ascospores. Here you can have the ascospores or 8 ascospores in each ascus. So biochemically in these reactions, a particular enzyme is not there, a particular biochemical pathway is suppressed. In the same way, a particular DNA, I think I told you already, DNA is nothing but a polynucleotide chain. It is a gene, and had to happen any change in the structure of DNA, it leads to the development of particular amino acid leads to particular protein leads to particular enzyme is not there, a particular biosynthetic pathway is not finished. So that there is a link that you can study in this Neurospora by giving some mutations or some chemicals. You can reduce a particular enzyme and you can see the particular biochemical pathway. Another example you have Physarum polycephalum. Physarum polycephalum is a very good research material for the study of DNA, DNA synthesis, morphogenesis, that is development of morphological organs is called morphogenesis, and mitotic cycle, cell division, etc. You can study with this material. Next, medicines. So the excellent, the miracle medicine, that is penicillin, that is developed by Alexander Fleming in 1929. It became a miracle drug, and the penicillin is also obtained from Penicillium notatum and Penicillium chrysogenum. And citronine, these are all antibiotics which help to kill the microorganisms, and citronine is obtained from Citrum Penicillium citrinum, and Graciofulvin from the Penicillium Graciofulvin, and Ketomine from Ketomum, that is Coccoioides, and ergotine from Claviceps purpurea, and clavin from Aspergillus clavatus. More than that, many medical, these what called antibiotics are obtained from different fungi. Next, we'll see the enzymes. So I told you already, the zymase is obtained from this is Saccharomyces or yeast. Many other things, these enzymes also obtained from different fungi. This talk of diastase, diastine, and polyzyme, etc. are produced by Aspergillus flavus. Aspergillus flavus. Aspergillus flavus. Amylase is from Aspergillus niger. Amylase from Aspergillus niger and Aspergillus oryzae. Zymase invertase from Saccharomyces cerevisiae. That is the botanical name of yeast. Botanical name of yeast is Saccharomyces cerevisiae. I think you are following. The next, vitamins. Vitamins, vitamin B complex and riboflavin are produced by Saccharomyces cerevisiae, that is yeast, and ergosterol, which contain vitamin D, is synthesized from a number of molds and yeasts. So ergosterol and vitamin D also coming from number of molds. Molds are common name for fungi, molds and yeasts. Regarding the hormones, gibberellin is a hormone. Gibberellin is a hormone that induces internodal elongation. Actually, in plant season, that is paddy vegetation, a farmer found out some plants are very tall. So he thought that there is more manures may be there in those plants, but later it is observed these plants are affected by a disease. that is called Bacchain disease. Later, a chemical is obtained from this diseased plant that is called gibberellin or gibberellic acid, that has a capacity to induce internodal elongation and sudden flowering of the plant. The technical term is there for that, that is called bolting. Bolting means internodal elongation and sudden flowering of the plant because of this gibberellin, and it is also having many physiological functions that is affected by the fungus Gibberella fusicori. Gibberella fusicori. The next one, fungi as food. So some of the species of fungi, since they are having vitamins and proteins etc., used as food. For example, mushrooms like Agaricus bisporus and Agaricus arvensis and Agaricus campestris, these are cultivated and giving food material to other places. Next thing is the biological control. So biological control, just like in bacteria we learned that Bacillus thuringiensis, the biological control to kill the caterpillars of plants. The same way here also some biological control are there. Some soil inhibiting fungi, that is a trichoderma, kills the Pythium fungus. This is one fungus. This is also fungus. It causes root rot disease in plants. Root rot fungus. For root rot and plant will die. So for that fungus is eaten by trichodermium, that fungus. Another example, Penicillium vermiculatum checks Rhizoctinia solanae. Another fungus, it causes disease in Solanaceae family members, Rhizoctinia solanae, that is checked by or eaten by Penicillium vermiculatum. So next we will see the dactylaria and zoophagous and arthrobotrytis etc. destroy certain nematodes and eelworms etc., which cause plant diseases. These nematodes and eelworms causes diseases, but the plant dactylaria etc., what happen? It kills this process. It kills those fungus, the animals. These dactylaria, zoophagous and arthrobotrytis or fungi, they kill the nematodes and eelworms which cause the plant diseases. Why? How? With this fungal hyphae forms into knots and they kill the animals. So let us see. Here you see the thing is, the white colored one are the fungal hyphae and the brown color, they are called these nematodes. So these nematodes are catch, and around the nematode it produces a hyphae, and so that the nematode cannot move from one place to another place, soon it will die. These are called the predaceous fungi. The fungi which kill animals are called the predaceous fungi. Next, mycorrhiza. This is very interesting to help the plants. Association of fungi with the roots of higher plants is called mycorrhiza. And this mycorrhiza, you can have a difference with the parasites. That the parasites, where happen, the parasite only gets the help, whereas the host cannot. But whereas here it is a mycorrhiza is also a type of symbiotic association where both are benefited. In this, the association of fungi with the roots of higher plants called mycorrhiza. The other day we discussed the mycorrhiza may be ectotrophic mycorrhiza and endotrophic mycorrhiza. Example, Rhizoctonia, Boletus, Tricoloma, Phallus, Glomus, etc. They help in absorption of water and minerals. Glomus is a very good example, whereas this one is an endomycorrhiza, that is, it produces its hyphae inside the host plant and helps in absorption of nutrients, especially the phosphorus from the soil. This is a very good thing. So these mycorrhiza help the plant in absorption of water and also the nutrients, especially the phosphorus from the soil. So far we discussed only the beneficial aspects, and now we see the harmful aspects. So in this we see the plant disease, human diseases and animal diseases. Now let us see the plant diseases. Plant diseases, a list of plant diseases are there, but here I gave a very important one so that you can remember the pathogen and also the plant which will be affected and name of the disease. Certain fungi are parasitic on several crop plants, causing disease and decrease the yield. So whenever a plant is affected, the plant cannot have this chlorophyll, so it will not produce more yield. So some of them are Phytophthora infestans, causes late blight of potato. So it is also, this causes late blight of potato is called Bengal famine. So once in Bengal, it complete all the potato vegetation is spoiled, and the many people, lack of food, they died. So this is the Phytophthora infestans. This causes the late blight of potato. Albugo candida causes white rust of cruciferous. On cruciferous plants, it causes a white rust, a powdery white powder on the leaves, and it will affect the yield. The next one, the Syncytium endobioticum, causes the black quart of potato. Inside the potato, you have a round, a circle of black colored thing is there, so that the weight of the potato is reduced and also the yield also reduced by this Syncytium endobioticum. Regarding these plant diseases, Erysipeae causes powdery mildew of cucurbits, the cucumbers plants. It causes a white powder disease on the cucurbits. Puccinia graminis causes rust in cereals. The other day I told you already, the Puccinia graminis, this is a heterogeneous fungi. It needs 2 plants to complete its life cycle, and it causes rust disease in cereals almost, in wheat and paddy, etc. causes disease. This Puccinia graminis causes the rust disease. Rust means red color, red powder disease. It causes red powder of leaves and it reduces the yield. Ustilago causes the smut disease in cereals. Smut means black powder disease. This is red powder is rust, black powder smut disease in cereal plants. It affects the inflorescence, leaves, and etc. and affect the plant. Politotrichum falcatum, I think you might have experienced in sugar cane. If you cut a sugar cane where you can find the red streaks or red spots are found, that is due to this Politotrichum falcatum. It causes red rot disease in sugar cane and reduce the what's called juice content and also yield. Another thing, Helminthosporium voriae, this causes the brown spot disease in rice and reduce the yield. Regarding the harmful aspects, the human diseases, one is the aspergillosis in skin by Aspergillus niger. So Aspergillus niger causes skin disease. In the diagram you can find some reddish patches are found in the cheeks of this person. So it is also contagious when it is irritating. Just if you use the nails, and if the same nail used in another place, it will be contaminated. So one place to another place. This is aspergillosis caused by Aspergillus. Another disease, ringworm. It is quite common in our area. That is a ringworm disease. So there the microsporum infect and having the patches are round in shape. It spreads throughout the body, spreading throughout the body. Even if you use the patient's dress or any cloth or handkerchief, etc., it will be contaminated from one person to another person. This is ringworm by microsporum. Another disease by automycosis in ear. In ear, you can have automycosis by Aspergillus nidulans. So many students are having a habit of just putting their pencil or pen in the ears. So that what happens sometimes, if any spore is there, it will be infected the ear, then the pus formation comes and damage of the ear also takes place if you ignore the disease. The next we have the dandruff. It is quite common among ladies and gents, the formation of this microsporum furfur. This is also contaminated from the one person to another person. If you use the same comb used by the dandruff person, it will be contaminated to other people. This is microsporum furfur. Another athlete's foot. Generally the athletes, what happen, they are having the socks and also shoes continuously without leaving, and where you can find some sweating is there, so that the infection takes place. And also this is also contaminating. If the same shoes or his socks are used by another person, the same disease is contaminated from one person to another person. These are found by in between the fingers of the leg, and sometimes in the hand also, by Tinea rubrum, that is the fungus name. The disease is known as the athlete's foot. The next one, let us see the diseases in animals. So in this human beings, I told you the ringworm patches are round. The same you can find in the animals also. So you can see a pig is there having full of this ringworm disease. This is caused by, in animals, it is caused by microsporum, epidomycophyton and trichophyton. So these are responsible to give the ringworm disease in animals. So in animals also you can find athlete's foot. So here this is an animal, leg is there, there you can find the fingers and toes. Then in between you can find the disease caused by Tinea rubrum. The next, in animals, aspergillosis. That is also having some patches, infection to patches found by Aspergillus species. Aspergillus species, here you can have a mouth of this dog. There you can find, around the mouth you can find the white patches are there. Those are the infection spots by Aspergillus species. Another is called penicillosis. That is another disease caused by the penicillium. So here you can find there is a horse. On the skin you have some patches. So that what happens, the skin, the hair may be removed slowly and the infection may be increased unless you take remedial steps. This penicillosis disease is caused by penicillium species. The next, spoilage of foodstuffs. So many food materials are spoiled by fungus. Any food material having a moisture, that is immediately infected by this fungus. So there is quite a normal in our houses, you can see on bread and also in a coconut pieces also you can find the infection comes from the fungus. So spores of Rhizopus, Mucor, Aspergillus, Fusarium, etc. germinate on jam, jellies, pickles, bread, etc. and destroy them. And penicillium, Rhizopus, Mucor, Aspergillus, etc. spoil the meat during the storage and the transportation, and the Rhizopus and Mucor, Aspergillus also spoil the fruits. Oranges, tomatoes, grapes, etc. are spoiled by the Rhizopus, Mucor. And destruction of goods, destruction of the materials you can find by many saprophytic fungus like Penicillium, Alternaria, Trichoderma, Mucor, Ketonium, Fusarium, etc. destroy leather, cloth, rubber, paper, photographic materials and optical instruments. In rainy season you can find an experience that in our shoes and belts also you can find the patches of the Aspergillus and Rhizopus and Mucor, they infect. and most of the paper also you can find, paper may be spoiled, or sometimes it may be stained by some dots on the paper, and optical instruments like the lenses of photographs and also cameras also spoiled due to this fungal effect. Wood decay. Generally, if it is a low standard wood, generally you can find a yellow powder, just a dust like thing is found in the wood, and certain saprophytic bacteria, fungi like polyporous attack wood and causing wood rot. Polyporous causes wood rot disease, and Penicillium, Fusarium, etc. cause stains on the wood. You can find some stains, red color, yellow color stains found on the wood. Main wood rot by the polyporous. That wood rot that you can find, this is two diagrams you can find where the wood is spoiled. Wood is spoiled, make into small pieces, and dust of these things also come in the attack of wood. And cancer causing fungi, you can find cancer causing fungi, you can have that aflatoxins or mycotoxins, this is OR, or mycotoxins are produced by Aspergillus flavus, Aspergillus fumigatus, etc. They cause liver cancer. So it is a general experience you can find in a groundnut. You can find any piece, groundnut pieces, dried groundnut pieces where you can find some yellow color that is poisonous. There you can find Aspergillus. So this is a poisonous substance obtained from Aspergillus flavus. Aspergillus A. flavus toxins, chemical substance, a poisonous substance. So it is obtained from Aspergillus flavus and named as aflatoxin. So these are produced by these Aspergillus species, and they cause cancer in animals as well as human beings. So regarding these hallucinogenic fungi, so this is some fungi like Claviceps, they contain LSD, lysergic acid diethylamide, which has hallucinogenic property. So that is distortion, a drunkard, what a drunkard feel, like that, a feeling of being happy, that's all. That is actually a permanent happy. That is the materials you can get this from Claviceps, etc. that they consume. What happen? Distortion of perception takes place for temporary. So these are some dangerous fungi. So in this way you can see the fungi, general characters, and reproduction, classification, and economic importance. Tomorrow we will learn remaining some life forms of this fungi, and a new branch or new subject, that is lichens. Thank you.